Oh, shit.
Okay, as compared to this. Okay, so I can ask you, what are this? What is the process? And I can ask you, what are those? And of course you know, that's your what? What are these little things here?
Sister chromatin.
Chloroplast.
We have your expression for you.
I got it.
This is your chloroplast, and what is this thing here?
Vacuum.
This one here? You can see the vacuum,
The nucleus.
the nucleus.
B is the nucleus.
What stage is that? Anaphase.
Anaphase.
You should know that right away. What stage is that? What
Metaphase.
stage is that? What stage is that?
What was it?
Or was what?
That was telophase, right?
Yeah.
Oh, okay.
Because you have the breaking of
And what stage is that?
Cross.
Prophase.
Prophase. Beginning of the spindles.
Prophase.
No one knows.
Beginning of the spindles.
Oh, yeah. So, man.
Quick picture.
She's, oh, man.
You guys are so funny.
So many questions.
Stop it.
Wow.
Stop the madness. There's more chance to get a lot more wrong.
Yeah.
Yeah.
It says
What?
I made the exam last year.
What?
Email it to me.
Oh.
And he'll make sure everybody gets it. He'll CC it to all of us.
I'll deliver it.
Smiley kid.
Personally.
Why didn't I been think of this? Why didn't I been do this?
Okay. Let's look at past questions. Let me go back to this.
Oh.
It had to be you. It could not be anybody else but you.
Yeah.
My goodness. Okay. Osmosis, Brownian movement, diffusion, lysis. Need to know those. Okay?
Oh, yeah, I can see that good.
The semi-permeable membrane, permeable membrane. What is your dialysis tubing called? Semi-permeable or selectively permeable membrane, right?
What's it?
Phenethylline.
I'm writing
So what?
Phenethylline is what? An indicator, right? What do you use it for? What do you use an indicator for?
this.
Phenethylline?
No.
No, the,
What do you use it for? What are you guys doing? Recording the whole thing? Wow. So why am I even talking if you're recording?
I'm recording the voice.
I love to hear your voice. I'm going to plug this to my auxiliary cord while I'm
That's right, it's the
driving.
last test. I just listened to you talk about the entire thing.
Really?
Yeah,
So
I
you gonna
can.
do well on the test?
Oh, yeah.
Keep talking.
Toy, you gotta send that to me.
Oh, I will, girlfriend.
Alright, because I can't see it.
Phenethylline is a pH indicator. Used for pH. Hypotonic, when the surrounding is lower than the
The inside.
Hyper is when the other way.
The out, yeah.
Outside is higher. ISO is when
it's the
two
same.
A cell in hypertonic will do what? Lose water, gain water. Hypotonic,
Gain water.
lose water, gain water.
Gain water.
Lose.
Hypotonic, lose water, gain water.
Or to lose water.
Hypo,
Hypo is when
it
it
will gain
swells.
water because the inside is higher than the
I thought
outside.
you said hyper. It says hyper.
I did say hyper?
Yeah.
I need hypo.
It's time to
Hypo
go.
is
Time to go.
I said hypo.
No, you said
I
hyper.
said hypo.
you said hyper.
I mean, it's recording. Do you really want to fight? You
No. Hypo will gain, hyper will lose.
Right.
Right.
Okay. Remember your red cells, your plant cell does not lose or gain. It goes to plasmolysis, right? All right. Enzyme. I don't know where these questions came from.
You made them up.
sorry.
Bless you.
Okay. Enzymes. Enzymes blank what by doing what? What is the purpose of an enzyme? Lower the activation rate. Energy. Okay. Enzymes does what also function for optimal condition, correct?
Yes.
The substrate that you did, remember the potato extract?
Yeah.
Which one will be the substrate here?
Substrate.
The catechol. It was the captain salt.
Which one will be the substrate?
The,
The potato extract.
okay.
Okay, the catechol will be the enzyme. Process that you see, aerobic respiration. What would be the setup for aerobic respiration? What did you do?
The lights with the plants is that one?
With what? Was in the test tube where you had the plant in there and you blew into it, right? Yes?
What?
You blew into the
Oh,
Tube.
yeah.
into phenol red and you turn into yellow.
Right.
And then you had the plant in there and the plant changed it back to red.
Right.
What
That's-
would be the setup for fermentation?
The moonshine.
Or the moonshine, or the test tube.
The,
The yeast.
the yeast in the what?
And you have two of the test tubes.
No, I won't give you that.
Look at
What's the other one that you use?
that's
The
right.
pipe.
Oh yeah, the crack pipe.
The
That
crack
didn't
pipe.
work.
Oh, the
That
crack pipe
didn't
thing.
work.
Fermentation is the crack pipe.
Yes.
Okay.
The crack pipe was. Better not put this on YouTube. I said, what kind of biology are you teaching this student?
You just identified
Chemical
in class.
biology.
We know what you're talking about.
Explain
I'm
it.
just trying. It's time to go home.
That's more important than the practice exam that she's giving us?
I would have let that rain,
I would
honey.
let that
I
go.
would kick him out of this class.
This is so mean.
And give everybody 10 extra points.
Photosynthesis. What would be the setup for photosynthesis?
Plant, probably.
You got
So with the
plant,
snails.
it's on the light.
The light on the plant, right? With the foil on it. All right. The gas that is collected depending on what is set up. If it's aerobic respiration, what gas are you collecting?
Carbon dioxide.
If it's photosynthesis, what gas are you collecting?
Sorry.
Oxygen.
If it's fermentation, what gas are you collecting?
Carbon monoxide.
Yeah.
Carbon dioxide.
No. Why would you guys do that?
Hey, you're
thought I
watching
had it
one
right.
video here. Why don't you,
I'm disagreeing because I was.
If it's fermentation, what gas are you collecting? Come on.
Carbon dioxide.
Carbon dioxide.
Okay, go one more time. Aerobic respiration is which
Carbon
one?
dioxide.
Aerobic CO2, photosynthesis O2,
Oxygen.
fermentation, alcohol, and CO2.
Okay.
Aerobic respiration, CO2, and what is it now? ATP, right? Photosynthesis, glucose and oxygen.
Right.
Fermentation, alcohol, and CO2. And we did not do photo respiration.
Right.
Okay? Say yes?
Yes.
All right. What do you use? If catalase is the enzyme, what did you use for the catalase? What did you use to break down the enzyme catalase? What did catalase break down? Which one of this?
The peroxide.
Hydrogen peroxide broke it down to what?
Water and oxygen.
Water and oxygen. You need to know that.
Which?
Huh?
What lab was
What
that?
lab?
What lab was that? The enzymatic lab that you did, Mama?
Some water.
You did it. I didn't do it.
Yeah, I didn't.
Some water.
Yeah.
It doesn't matter. Your group did it.
I know, I don't remember.
What does it break down?
What?
What did you say it breaks down?
Hydrogen peroxide
Yeah.
to water and oxygen. What? Who did it? Abundance, you did it?
It breaks it.
You need to explain it to her?
Water. It breaks it down.
Funny.
I can't believe that, y'all.
All right. The only thing that is similar here is the glucose, which you will see in. I don't even know what the station was about. Oh, it's time to go home. Glucose you see in what? Photosynthesis, right? Ethanol you see in fermentation. What causes this reaction? What could it possibly be? What reaction will this be if you look at the answers? The yeast. Yeast is in what? What process?
Fermentation.
Fermentation.
Can all that question just be about making beer and alcohol?
You did germinate. No, you didn't do that.
Germination? No.
Okay, ethanol, CO2 is the same thing we just talked about. You did your KOH, right? KOH was your what? What experiment was that?
Oh,
The
with
what?
the peas.
The peas, right? Respiration in peas, right?
That didn't work out
And the
too well.
KOH was for what? What was it to do? What was the purpose of the KOH? To absorb the excess CO2. Okay? I don't know what that is.
That one.
Well, this one you already know the difference, the three of them. Reaction as a CO2 blend, the color changes to, yeah, that's it. That's a phenol red that you did. So as the reaction proceeds, CO2 is what? Release, and the color will change to red. CO2 is liberated, and the color will change to
Liberated.
red.
They come to say release.
Okay? I don't know what that is. Carbonic acid. This is the phenol red. It was red, phenol red. You turn it into yellow. So you turn the solution from base to carbonic acid to yellow. So what is the name of the solution in tube A after this process? The process is aerobic respiration, right? Yes?
Yes.
Say yes.
Yes.
The table. Somebody's at the table. Quentin? She's talking to you.
I know, I'm trying to stay still.
You can stay. You
I
can.
can't sit still. I'm dead serious.
I know you're serious. I'm going to tie you down.
Oh my God, this is all on
Yo,
video.
hey, don't, we're in class. You can't talk about them steps in class. Anyways, it took me
This
serious.
is all on video.
All right. I'm going to give you something similar like this, but I'm not going to ask you the whole thing. This is basically what? What is this
Paper
represent?
chromatography.
Paper chromatography. What did you use? The pigments, right? Plant pigments.
Plant.
So you're looking at the separation of the pigment in the plant. Where you have your chlorophyll and your keratin and your xanthophyll. Okay? Paper chromatography separate pigments on the basis of their
Solubility.
and the RF, of course, I can ask you, give you that paper for you to measure the RF. And the RF is what? Reference? Front? Will be from where? Point of application to
Distance.
The end.
the distance, the end of it. Okay. The liver extracts. I don't think you guys did that.
No.
The cell cycle. You gotta know your
Yeah.
interface. Know your prophase, know your metaphase, anaphase, and telophase.
That's my anatomy test.
Okay? Know the difference between the stages, especially the last stage in plants and the last stage in animal, which is basically your cytokinesis. Okay, there's no cytokinesis in plants, rather it's cell plates. And again, identify those things. In the cell organelles, you know all this stuff. You know what a mitochondria looks like and chloroplast, right? Nucleus, yes? Okay. Chromosome, bacteria. You should know what a bacteria looks like. Of course, you should know what a sperm cell looks like, and you should know what a chromosome looks like.